everyone and welcome to Miss Estruch Biology. In this video we're going through the entire topic 5. So if you do need to either prepare for the topic, revise for it, then this is for you. Now if you are new to this channel, then hello, I'm Miss Estruch. I've been teaching for over 10 years at schools and now I also teach on YouTube and create lots of resources as well. So if you do want that extra bit of help in biology, then make sure you subscribe so you don't miss out on any of the latest videos. Thank you so much to all of the lovely messages that you've been sending me on Instagram at the moment. Here are just some of them. So many of you are just getting your mock results back at the moment, and it really makes me happy when I see that you're doing so well in your mocks or that you've made huge improvements since using my videos. And also so many of you have said that using my notes have helped you to get A's and A stars in the mocks. So that is fantastic. Now my notes, if you do want to get your own copy, this is what they look like just here, and I'll link it in my description so you can get your own copy. They contain all of the theory for AQA biology. It highlights the key notes, the key marking points to help you with exam technique, as well as essay links, which is one of the harder parts of the AQA exam. So if you also want to boost your grade up to an A or an A star, then give them a go. But for now, let's get into topic 5. So the first thing to do with photosynthesis is knowing where it occurs and just a mini recap on the structure of a chloroplast, because you need to know where each stage of photosynthesis happens. So here on our chloroplast, we can see the thylakoid stacks, and the thylakoid membrane is on the outside of that. And that's where you have these folded membranes where you have all the proteins embedded for the electron carrier chain, and also ATP synthase. And that is where the light-dependent reactions occur. A stack of them is called a granum. The fluid part in the centre is called the stroma, and that's where the light-independent reactions happen, and you have lots of enzymes there for those. And then we have the inner and outer membranes as well, which are controlling what can enter and exit. Photosynthesis is a two-stage reaction. We have the light-dependent and the light-independent reactions. And the light-dependent reactions happen first, and as the name suggests, it requires light. This happens on the thylakoid membrane, or you could call that the granum. In this light-dependent reaction stage, light energy is used and water to create ATP and reduced NADP. And those two products are required for the light-independent stage, which comes second. So there's 4 key steps here. We have photolysis, or sometimes it's pronounced photolysis to remember the stage, photoionization of chlorophyll, and chemiosmosis. Now those are actually the 3 processes. Production of ATP and reduced NADP is the outcome, but AQA states these are the 4 key stages that you need to know. So starting with photolysis or photolysis, photo refers to light, lysis means splitting. So photolysis of water means light energy is absorbed and then that splits the water. And it splits it into oxygen, electrons and protons, which are hydrogen ions. What then happens is the protons are picked up by NADP to form reduced NADP or NADPH, and that is then used in the light-independent reaction. The electrons are passed along a chain of electron carriers, and the oxygen is either used in respiration, but it's not used in photosynthesis. So if it isn't needed for respiration, it will diffuse out through the stomata. We'll go through in a bit more detail what happens to the protons and electrons when we get to chemiosmosis. Photoionization of chlorophyll, again, is to do with light, because we've got photo. So light energy is absorbed by the chlorophyll, and the ionization part is referring to the fact that the light energy results in the electrons gaining energy. And we describe that as them becoming excited and raising up an energy level. And that causes the electrons to leave the chlorophyll, and therefore the chlorophyll has been ionized. Some of the energy from the released electrons is used to make ATP and reduced NADP in chemiosmosis. So let's have a look at chemiosmosis then, because this then links it all together for the light-dependent reactions. So we've said that the electrons gain energy from the chlorophyll, will be released from the chlorophyll. What happens is those electrons move along a series of proteins embedded within the thylakoid membrane. And as those electrons move along the proteins, they release energy. And some of that energy is used to transport or pump those protons from the stroma across a protein and into the thylakoid lumen. This then results in a concentration gradient being built up, but we actually call it an electrochemical gradient because we have a charged molecule, so we have ions there. So now we have this electrochemical gradient that enables the protons to move by facilitated diffusion back down their concentration gradient to the stroma. But the only protein that they can attach to is ATP synthase. And as the protons attach and move through ATP synthase, that enables the enzyme, which the protons move through, to phosphorylate the ADP into ATP. So that's how the ATP is produced. We then have the protons back on the stroma side, and this is how we create NADP that is reduced, or reduced NADP. So the NADP coenzyme picks up the electrons from the end of the electron transport chain. It picks up the protons after they've passed through ATP synthase, and that reduces NADP, which makes it NADPH. So now we've created ATP and reduced NADP. Those two molecules are used in the light-independent reactions, also known as the Calvin cycle. This occurs in the stroma. It requires enzymes, and the enzyme Ribisco is the enzyme that you need to know. And because of that, this stage is temperature sensitive. But it doesn't require light energy for the Calvin cycle to occur. Now, the Calvin cycle uses carbon dioxide as well as the two products from the light-dependent reactions, and we create a hexose sugar. The ATP that is needed is hydrolyzed to provide energy for the reaction, and the reduced NADP is used to donate the hydrogen to reduce the molecule GP within the cycle. So let's go through this cycle then. The first part of the cycle we'll look at is how the carbon dioxide enters the cycle. So carbon dioxide reacts with RuBP, and that is a 5-carbon compound. So all of these yellow circles are representing carbon molecules. So we can see we've got a 5-carbon compound reacting with carbon dioxide, which only has 1 carbon. That would result in a 6-carbon compound, but actually it's unstable, so it splits into 2 3-carbon compounds, and that's our GP. So we have 2 lots of GP. This is then where one of the ATP molecules is used, and it's where the reduced NADP is used. GP picks up the hydrogen, and therefore NADP is reformed, and the GP is now reduced to form TP. That reduction requires energy from ATP. The TP is then used for 2 things. One of the carbons is removed from these 2 times 3 carbon compounds, and it goes towards creating a hexose sugar. That will leave behind 5 carbons, but as a 3-carbon compound and a 2-carbon compound. So ATP is needed to form those back together, or in other words, regenerate RuBP. Now, that is just one round of the cycle, and that only created 1 carbon, which isn't enough for a hexose sugar. Hexose means 6. So the Calvin cycle has to happen 6 times before a hexose sugar is made. And that could be glucose, it could be sucrose, and then the plant can use that glucose or sucrose and convert it into whatever organic compound is needed. So that could be another carbohydrate, so cellulose for the cell walls. It could be a store of glucose in the form of starch, or it could convert the hexose sugar into glycerol and then combine with fatty acids to make lipids, or it could combine it with nitrates to form the amino acids. Now you need to know a little bit about limiting factors as well, and the limiting factor is anything that reduces the rate of photosynthesis, and that could be light intensity, carbon dioxide concentration, or temperature. And the way you can look at a graph to tell what is currently limiting the rate of reaction is wherever you have a positive correlation on the graph, that tells you that whatever is on the x-axis is currently limiting the rate. So that means up until this point, or probably a bit further up to here, light intensity is limiting the rate of reaction. And we know that because as we increase the light intensity, the rate increased. But that then starts to plateau and level off. So even though you add more light, the rate doesn't increase. And what that means is there must be another factor that is now limiting the rate. And that must be either carbon dioxide concentration or temperature. So it's the same concept with carbon dioxide concentration, that as the rate is increasing and the carbon dioxide concentration is increasing, we know that carbon dioxide is the limiting factor until it plateaus. Temperature, this is linked to the idea of enzymes because it's an enzyme controlled reaction. So at low temperatures, the rate of reaction is lower because we don't have enough successful collisions. There's not enough kinetic energy. We don't get enough enzyme substrate complexes. But if it gets too hot, the enzyme denatures. Now the carbon dioxide, if you had to say why that limits, that is because it is a reactant in the Calvin cycle. Light intensity can limit the rate of reaction because light energy is needed in the light-dependent reactions for photolysis and photoionization. So for maximum photosynthesis, in agriculture, when they're growing crops, they need to consider how they can remove those limiting factors to maximise their profits. So sometimes you get application questions linked to this. So the idea of adding in artificial lighting, heating a greenhouse, and that could be through burning fuels because that will also produce carbon dioxide. But all of those additions cost. So it has to be considered whether the cost of the additional light, heat or fuel is offset by the additional profit. So they have to make sure it's cost effective in order for it to be worthwhile to pay for those additional conditions. Next then we have a look at respiration, starting with aerobic respiration. And there are 4 key stages: glycolysis, the link reaction, Krebs cycle and oxidative phosphorylation. And for each stage, you need to know exactly where in the cell it occurs. And that's what we have in brackets here. So we'll start with glycolysis. This does produce some ATP, but not a lot. And it also happens in anaerobic respiration because oxygen is not required for glycolysis. It happens in the cytoplasm, not actually inside of the mitochondria. And there are 3 key stages that AQA say you need to know. First thing that happens is that glucose is phosphorylated to glucose phosphate, and that uses 2 molecules of ATP. That glucose phosphate very quickly is converted into triose phosphate, and then triose phosphate is oxidated to form pyruvate, and that produces 4 ATP and reduced NAD, which is the coenzyme used in respiration. So let's just have a look at what that would be on a diagram. Again, the yellow circles are representing carbon. So glucose is a 6-carbon compound. We said the first thing that happens is glucose is phosphorylated, and what that means is phosphate groups are added to it. Now we're going to add 2 phosphate groups, so we need 2 ATP molecules, and that creates glucose phosphate. That is an unstable compound, so it splits into 2 3-carbon compounds called triose phosphate. And then the triose phosphate, or TP, both of those molecules are oxidized. Both TP molecules will result in the gain of 2 ATP when pyruvate is made, and also as each is oxidized, there'll be a reduced coenzyme created. So NAD picks up a hydrogen to form NADH, or reduced NAD. So at the end we have 2 molecules of pyruvate, a net gain of 2 ATP, and what we mean by that is, although we've made 4 ATP molecules in this stage, we had to use 2 at the beginning. So overall, we've created 2. we've also made 2 reduced NAD. So that pyruvate that has been created and the reduced NAD, they are needed in the next stages, which is within the mitochondrial matrix. So they have to be actively transported into the mitochondrial matrix, ready for the link reaction. So the pyruvate is going to be oxidized further to make acetates. We're going to have an NAD picking up a hydrogen to create yet another reduced NAD molecule. So that's the first step we can see here. Pyruvate, our 3-carbon compound, it's going to be oxidized further, and we can tell it's been oxidized because it's lost the hydrogen, which has been picked up by the coenzyme NAD. So that has created acetate. Acetate is only a 2-carbon compound, and that is because carbon dioxide is produced at this stage of the link reaction. Now that's not the end, because the acetate then combines with another coenzyme called coenzyme A, and that creates acetyl-CoA. And acetyl-CoA is what can then enter the Krebs cycle. So overall, for every glucose molecule, there are 2 pyruvates created. So for every glucose molecule, the link reaction happens twice. So for 1 glucose molecule, we'd create 2 acetyl-CoA, 2 carbon dioxides and 2 reduced NAD. So that acetyl-CoA can then enter the Krebs cycle, and it combines with a 4-carbon molecule, and that will then create a 6-carbon molecule. Now the coenzyme, its only job is to deliver the acetate to that 4-carbon compound. So once that happens, the coenzyme is released and it can be reused in the link reaction. We then have a series of redox reactions, and the Krebs cycle is going to be generating even more reduced coenzymes, a small amount of ATP and some carbon dioxide. And that's what we can see in this stage here. The 6-carbon to the 4-carbon molecule, which is then regenerated. To go from 6 to 4, we must have lost 2 carbons, and that's in the form of 2 carbon dioxides. We only have 1 molecule of ATP being produced, but we have quite a few reduced coenzymes. We create 3 reduced NAD and 1 reduced FAD, which is our final coenzyme involved in respiration. So overall, the products per cycle are 3 reduced NAD, 1 reduced FAD, 1 ATP and 2 carbon dioxides. But for every glucose, the cycle happens twice because we'd have 2 acetyl-CoA produced, so 2 rounds of the cycle. So we actually double all of those values. So look carefully at an exam question, whether they're asking for the products per cycle or per glucose molecule. Oxidative phosphorylation is the final step, and this is very similar to what we looked at in chemiosmosis in the light-dependent reaction of photosynthesis. It's when most of the ATP is produced. So the first thing that happens is, in the mitochondrial matrix, all of those reduced coenzymes that were produced are going to release their hydrogen, and that splits into protons and electrons. Those electrons are going to be transported along the electron transfer chain. That releases energy, and that energy is used to transport the protons from the mitochondrial matrix into the intermembrane space. That creates this electrochemical gradient, and therefore the protons can move by facilitated diffusion down their concentration gradient through ATP synthase. And the ATP synthase will then phosphorylate ADP to create ATP. So that is how we create lots of ATP in this stage, and you actually create 34 ATP molecules. Now at the end of the electron transfer chain, those electrons are picked up by oxygen. And oxygen also picks up the protons once they've passed through. And that is how we create water. So we say that oxygen is the final electron acceptor, and it will form water when it also picks up those protons. Anaerobic respiration is without oxygen, and it only occurs in the cytoplasm. In plants and microbes, anaerobic respiration produces ethanol and carbon dioxide. In animals, it produces lactate or lactic acid. And this stage is all to do with reoxidizing NAD so that glycolysis can continue to occur. So what we have happening is glycolysis occurs as usual because no oxygen is needed for that stage. But instead of the pyruvate being transported into the mitochondrial matrix for the link reaction, it remains in the cytoplasm and the pyruvate is reduced to form lactate. We know it's reduced because the reduced coenzymes that were just produced in glycolysis are immediately reoxidized to NAD. And that is because the pyruvate gained the hydrogen. Now that is important because that NAD can then be reused in glycolysis so that glycolysis can continue to occur. And that ensures that at least some ATP is continually made. So the downside is lactic acid is acidic, so it can denature enzymes and other proteins, but that is the price to pay so that at least some respiration can still occur. But you can't respire anaerobically for long because that buildup of lactic acid will eventually denature the enzymes involved in glycolysis. Same idea for microbes, except we have ethanol and carbon dioxide produced. Again, ethanol is going to be harmful, but it's to ensure that glycolysis can still continue, so at least some ATP is being produced for metabolic reactions to continue. The next bit in this topic is looking at how energy is transferred in an ecosystem. So it's looking at food webs. And all food webs begin with producers, which are green plants because they can photosynthesize to use the light energy to make their own organic compounds. So they don't need to eat, they produce their own food. Between each trophic level, which is the term of what we call each stage in a food web, the majority of energy is lost, and that is due to respiration and excretion. The remaining energy is used to form the biomass. And the biomass is the mass of carbon within the organism, or you can describe that as the dry mass. Now that is a key term, so definitely learn that definition. And we give biomass per given area, so it can be a fair comparison between organisms. NPP and GPP are two key terms that you need to know. So how productive an ecosystem is depends on the abiotic and biotic factors that are within that ecosystem. If there's plenty of water, light, it's warm, there's lots of green plants, then you'll have a really high rate of photosynthesis, and that results in more carbohydrates being produced by the plant. And we can quantify this using GPP and NPP. So GPP is gross primary production, and it is the chemical energy stored in the plant biomass in a given area, or it could be volume, and it's the total energy resulting from photosynthesis. NPP is the net primary production, and that is the GPP minus the energy that will be lost by respiration. So basically NPP is the energy you have left to go towards making the biomass. Another formula that you need to be aware of is this one here, and this is looking at the net production of consumers. So to work this out, which is represented by the letter N, for organisms such as animals, you'd use the formula below, where N is the net production of the consumers, I is the chemical energy stored in ingested food, F is the chemical energy lost to the environment in feces and urine, and R are the respiratory losses. So here's the formula, and it's come up a couple of times as just a multiple choice question, just ticking which is the correct formula. So that's a good one just to learn, maybe put on a flashcard. So here's just a visual representation. We're basically looking at the amount of energy going in minus the energy losses coming out, which is the energy lost in respiration and the energy lost in feces and urine. The rates of productivity are recorded using the units kilojoules per hectare per year. So there's 3 parts to those units, and it can be asked as a 2-mark question to give the units or to say why you have 3 parts to the units. So the kilojoules is the unit of energy, but these units also include per unit area and per year. So it's recorded per unit area to standardise the results to enable environments to be compared. So it's taken into account that different environments will vary in size. The units are also per year to take into account the impacts of the seasons. So when you have rain, when it's light or when it's less light, the temperature changes. That's going to affect the productivity because it affects photosynthesis. So we do it per year, so you have an annual average, so it's a fair comparison. The last thing within this topic is the nutrient cycles. And we're going to start with the nitrogen cycle. The nitrogen cycle is really important because nitrogen is within proteins, ATP and nucleic acids. So that's the DNA and RNA. So it's essential that animals, plants, microbes have nitrogen within them to create these molecules. And we need this cycle because you can't actually gain nitrogen gas through gas exchange like you can with oxygen or carbon dioxide for plants for photosynthesis. And the reason for that is nitrogen gas has this triple bond and we don't have the enzymes to be able to break those bonds and then use the nitrogen in compounds. Microorganisms do though, so they have the enzyme to be able to do that, or some of them do. And therefore we need this cycle involving microorganisms to make the nitrogen accessible for nitrogen containing substances, which plants and animals can then use. The nitrogen cycle involves these key processes: ammonification, nitrification, nitrogen fixation and denitrification. But you also need to be aware that saprobiotic nutrition and microbes are essential for all of those stages highlighted in blue. So saprobiotic nutrition is referring to when microbes are feeding on dead plant matter or waste, and it's the microbes that are going to be doing that. So if we have a look at the cycle, we have nitrogen gas in the atmosphere. There'll be nitrogen fixing bacteria that can convert that nitrogen into ammonia. And that will either be done directly by nitrogen fixing bacteria in the root nodules of leguminous plants, which are types of plants like clover and beans, which have these lumps or nodules on their roots that contain bacteria. So for them, the bacteria will directly convert the nitrogen in the atmosphere into ammonium into their roots. Plants that don't have those root nodules, there is free living nitrogen fixing bacteria within the soil, which can also fix nitrogen into ammonium. Then we have nitrification occurring, which is when nitrifying bacteria can oxidize ammonium into nitrites and then oxidize it further into nitrates. And that can then be absorbed by active transport into the plant roots. And then animals can absorb that through digestion and absorption. Now, the decomposers will be breaking down the nitrogen containing compounds in the dead plant and animal material and in the waste produced by animals. But for A level, it's not enough to just say the dead material or waste. You have to name the nitrogen containing compounds. So the waste, you could name urea. And then in the dead plant and animal material, it would be proteins. It could be DNA as well. So you have to name the compound that the decomposers would be breaking down. And this would be our saprobiotic nutrition. So the decomposers will break down the proteins to release the nitrogen, and it converts it back into ammonium, and then the cycle can go round again. If, though, there is a lack of oxygen within the soil, we then have anaerobic denitrifying bacteria taking over, and they can convert the nitrates back into nitrogen gas. So that's how we get the nitrogen going back into the atmosphere. So other than the denitrifying bacteria stage, all of the other stages are oxidation, and they would require oxygen to be present. The other cycle you need to know is the phosphorus cycle. And phosphorus is really important because it's used in the phosphate groups of DNA, RNA, ATP, and in the phospholipids within the phospholipid bilayer. So it is an essential element for life for all organisms. The phosphorus cycle is different to the carbon and nitrogen cycle, not that you actually learn the carbon cycle at A level, and that is because you do not find phosphorus as a gas in the atmosphere. So instead, the main store or source is as phosphate ions in the mineral form in sedimentary rock. Now you need to know about the mycorrhizae, which are fungal associations which occur between the plant roots and beneficial fungi. Now, although we're talking about this here in the phosphorus cycle, it would also be occurring in the nitrogen cycle as well. The mycorrhizae are going to provide benefits for plant growth. And that's because the mycorrhizae fungal associations, we can see here you get these really long networks of fungi, and they can spread out in the soil to create a larger surface area for the absorption of water and mineral ions going into the roots eventually. The plants provide an advantage to the fungus because the plants through photosynthesis will create carbohydrates and it can share those carbohydrates with the fungus. So this is a mutualistic relationship because they both provide a mutual benefit to each other for survival. So the phosphorus cycle, if we start in the middle looking at the phosphate ions in the oceans, or it could be within the soil, plants can absorb those phosphate ions through their root hair cells by active transport. Animals will then be eating the plants so they can consume and absorb the material to get the phosphate ions. When animals excrete, that will release phosphorus back into the oceans or the soil in the form of phosphate ions. As well as returning it to the soil, we also have decomposition. And phosphate ions from waste and remains are going to be broken down. So animals which have a shell, but also bones, contain phosphate ions. And those can be eroded to release the phosphate ions back into the ocean or the soil. The other thing you can have, which we can see in this image, is guano. And guano is from bird feces, and it's this white layer you can see on the rocks. So it's a type of excrement which is really rich in phosphate ions. Deposition is when we get phosphate ions building up to create the rocks, and that also links to sedimentation over here. So over time, from high pressure as well, the phosphate ions within the oceans, within the soil, will compact through sedimentation to create the sedimentary rocks. And then the last link back is the fact that those sedimentary rocks can be eroded to release the phosphate ions back into the soil or oceans. Now the extra one added here is the idea of fertilizers. Fertilizers added to soils can also run off from the soil or leach into water sources, so it would return it back into oceans, or it could just be within the soil itself, but that will be adding phosphorus into the cycle. If we just look at fertilizers a little bit further, we said that they can be added to the soil. The reason for that is when crops are harvested, you're removing the plants, and that is therefore removing one of the stores of nitrate or phosphate ions. So it's going to be taken out of the nutrient cycle. And if you continue to do that, eventually you'll have plants and soil which are mineral deficient. So you have to add either natural or artificial fertilizers to replace the minerals and the nutrients that are being removed when you harvest crops. So natural fertilizer would be animal manure. Artificial is soluble chemicals which contain exactly the proportion and quantities of the mineral ions required. Now, the natural fertilizers are cheaper because if you have your own animals on a farm already, it's completely free. It is just the manure from the animals. However, it doesn't contain necessarily the exact proportions of the minerals you need, and you have no control over that. Artificial fertilizers, because they are synthetically created, they are created to have the exact proportion of minerals that plants would require. However, the inorganic substances are very, very soluble, which has the downside of that they can dissolve in the rainwater and leach, which means they're washed away. So those soluble chemicals such as nitrate, phosphorus, dissolve in the water and then they leach, which means they run off from the rainwater into nearby water sources. such as rivers or ponds. If that nitrogen fertiliser leaches into waterways, it causes eutrophication. And eutrophication is when nitrates are going to stimulate the growth of algae in a pond or a river. It's more likely to happen in a pond though, because in a river the movement of the water can prevent the algae from growing or staying in one place. So the excessive growth of algae creates this blanket on the top of the water. That blocks the light. As a result, plants below that blanket aren't going to be receiving light for photosynthesis and they end up dying. Bacteria within the water will then feed and respire on the dead plant matter, and in doing that they're going to be using up the oxygen within the water, and you get more and more bacteria growing and thriving because of all that dead plant material to feed on, so even more oxygen is used up, and eventually there won't be enough dissolved oxygen within the water for animals, and animals such as fish and other aquatic organisms will therefore die because there's no oxygen for respiration. So that is it for topic 5. If you do want to learn it in more detail, then click on the playlist here so you can see each topic explained in more detail. If you have found this helpful, please give it a thumbs up and good luck with your studies.